Welcome everyone. My name is Michael Pratt. I manage the Technology Development Office at Boston University. If you're unfamiliar with our office, we manage the intellectual property for both Boston University and Boston Medical Center that include patents and copyrightable software and things we look to license to other parties. We also manage the licensing of those intellectual properties. And we support the BU community and the Boston Medical Center community through a number of different programs in addition to the patent filings that Rana runs to introduce people to entrepreneurship and moving their ideas forward. And you can reach out and contact us anytime. The shortcut to our website, the secret shortcut is bu.edu/OTD, which used to be our old website, but that gets you right directly to our website if you have any questions about that. I'd like to thank you for joining us today to celebrate innovation. And I mean that in a big context. I think innovation, when we think about it, moving towards products and services that help society, it's sort of a third agenda or a fourth agenda for the university mission. And it's important for us to celebrate it, step back and appreciate the hard work that goes into that. Today, we're going to celebrate in two parts. The first part, we're going to have a panel presentation organized, moderated by Rana Gupta, of some current and former BU innovators to share their stories and talk a little bit about that lab to market transition and the difficulty of shifting from a research mode to a product and commercialization mode. And then the second part, I'm going to introduce Gloria Waters, Associate Provost and Vice President of Research, to present the Innovator of the Year Award to Professor Ünlü, which I'm looking forward to. A few just small logistics. If this is a Zoom meeting as opposed to a Zoom event, so if you could keep your microphones on mute if you're not the speaker, that would be appreciated, because we'll hear all the background noise otherwise or disturbances. Also, submit any questions in the chat if you'd like. We're not going to answer the questions in real time, but we've already prepared answers for the questions that were submitted in advance, and we intend to take all those questions from the chat and post the answers to those questions along with a video recording of this event, so that if you want to go back and refer to what someone said, you can get access to the recording plus answers to any questions that you might have. So thank you for that. Now I'm pleased to welcome my colleague Rana Gupta to moderate the panel. Rana?
All right, thank you, Mike. Hello, everybody. I don't know about in your screen, but in mine, I'm in the top left. So welcome to our second Researcher on Tap series, because it's a focus on the researcher in my ever-present effort to impress upon people. It's people over ideas. It's the people that make the difference. It's people that create impact. So we'll hear from a few of our people. What we're going to do today is we're going to hear from 4 BU spinouts at different stages in their journey. Common to all 4 of these are the fact that a post-grad is leading or led the charge. So some of them are ongoing and some of them, one has spun out and now still being led. I asked the 4 teams in advance to answer the same 6 questions in their opening presentations. Some of you may recognize a couple of those questions because I'm pretty much a broken record when it comes to them. But the 6 questions in their opening presentations that the 4 teams will address are as follows. First, I said, introduce yourself. Who are you? Second, I asked them, what's your objective? And by that, I mean you personally. What's your objective in your heart? Not what's the objective of the idea, what's your objective? Third, what's the need for your idea? That is to say, who needs this? Fourth, oh, by the way, what's the idea? 5, where are you with your project? Where along the spectrum and the maturity? And then last, I said, look, you guys are going to have a platform. If you could shout out anything to your colleagues in the BU community about what you've learned and what they should know about what it means to try to spin something out the way you are doing it, then here's your chance to do it. So what's that learning? What's that one thing you would like to shout out to the community? So that's how they'll end each of those. So we'll start with Nick Vickers and Professor Sean Anderson. Then we'll move to Euros and then Josh and George. I'll let them do that. I'm going to shut up now and I'll come back on when George Daboul is finished. But with that, I'll say Nick and Professor Anderson, fire away.
All right, thank you very much. So sharing my slides, hopefully everyone can see them okay. So we'll just jump right into the sequence of questions that we were asked. So Nick, you want to go ahead and start off introducing yourself?
Sure. I'm a sixth-year mechanical engineering PhD candidate. My expertise is in optics and controls, and I've worked in 3 startup companies previously. And my main interest right now is in technology adoption, so getting something out into the marketplace and into people's hands. Okay, now Sean.
Right, so I'm a professor in both mechanical engineering and systems engineering. Background and expertise is in control theory, estimation with some robotics as well. And I mentioned a ton of things, of course, as all of us are, but relevant to today is trying to take some of the stuff that I've been working on over the years and transition it out, to transfer it out, to have an impact in a different way that I've been having so far. And there we go. So Nick, why don't you go ahead and tell us your objective?
So my main objective is to have a positive impact on society. I know that's kind of bland, but you know, when I was in high school, my mom almost died of a rare disease. And so I really wanted to make sure that other people that are in my shoes have a chance to have a positive outcome. Secondly, I want to see, I saw the power of Sean's research, and I want to get it into the marketplace, get impact beyond publication. And now that I'm at the end of my PhD career, it's a great time to kind of take that leap and take on that risk to try something new.
Right, and so for myself, you know, it's also a good time in my career to try something new. I wouldn't say I have things figured out, but I kind of know what I'm doing on the research side. I've been working on this project we're talking about today for quite some time. And it's just a great time to try something new with it, to try to have an impact in this different way. And I do want to have that impact. That's one of the 2 main goals for me here. You know, it's complicated stuff that we work on. Papers are great, but people are going to have trouble using it if we don't spin it out into something that they can get their hands on and apply in their labs. But also, and just as important to me, is Nick has passion here, and I really want to support him as he's going through this process. It's really exciting for me to kind of see him want to push this forward and carry this, and so I'm kind of happy to go along with him as we go through this ride. So let me talk just a little bit about the problem, the need for what we're trying to do. So we're interested in looking at dynamics in nanometer scale biomolecular systems. So our target potential customers are people like biophysicists and medical researchers, pharmaceutical developers. These are people who want to look at dynamics at the scale. They have questions they want to answer, but they can't get them right now. There's these barriers that are in the way. And these kinds of barriers are low resolution tools. So you can get kind of high resolution in space, high resolution in time, but it's a little hard to get resolution in both of those. They have limited measurement modalities. You can kind of see only one thing at a time when we really want to see lots of things at a time about what's going on. There's slow data acquisition, which is related to the spatial temporal resolution, but it is limited in some other ways. And finally, there are solutions out there, including the one we built in our own lab, but these tend to be very complex and bespoke instruments that require a fairly multidisciplinary team to pull together and to make work. And it's just not accessible to many of the researchers that are out there. But because of those existing solutions, we know that if we can get this to work and get this in the hands of the many, it's going to enable exciting science breakthroughs and exciting new understanding of disease mechanisms and things that we can't even dream of right now. So I'm really excited to see this get pushed forward. Which leads us, if I can figure out how to steer here, it leads us to where are we? So I started the ideas that we've been working on way back in 2008, soon after I got here. And I've been on this treadmill of getting the grants and doing the publications and getting the grants, doing the publications, which is great. And that's led us forward to having this working prototype in the lab. But this, I see, I skipped forward over the idea, but that's okay. So the idea is right here. So the working prototype is to track individual single biological macromolecules while also probing them to get additional information out and collecting information about their contextual environment. So in this picture, we've got this context down here, which is in this image is a neuronal cell. We've got tracking of individual molecules, which things are moving around inside this environment. And then we can look at them with a second source, if we like, to get information out about the dynamics inside these molecules, what's going on. And we've shown sort of each of these pieces, but we need to work forward to getting a working prototype with all 3. which leads us to where we are now. Nick has done the Spark program. So we're doing some customer discovery and primary market research. And as we're doing that, we realized that, oh no, we forgot to do a tech disclosure. So we had to take a step back and solve that issue, which we've done now. And so now we're pushing forward to hopefully get an ignition proposal, ignition award out so that we can fund moving ourselves forward to develop this full prototype. Once we built that prototype and have it working, we can take it out to the alpha users to get that initial feedback on what's really important beyond what we already understand. And then from there, move forward to commercialization. So that's kind of where we are. And that leads us to Rana's final question, which is the lessons we've learned in this process so far that we want to share. We're overachievers. We don't have just one, we have several. So Nick, there we go. There's the first couple. Nick, why don't you go ahead and start with the first one?
Yeah, so one of the most important things is that getting that tech disclosure in. If you don't have some sort of patent, especially for a lot of the technology we do, you don't have a startup. And so it's harder to launch it into the marketplace. And the second thing is, I had a great experience with iCore. My teacher, Beth Kahn, really put me through the ringer on trying to figure out how to actually talk to customers. And that part was new to me. I've never had to talk to people without talking about the tech, right? Focusing on their need. And so that was a paradigm shift for me, at least. Sean, take it away.
Yeah, so I want to just sort of echo those two things that Nick said. So the first is, the technology disclosures is not something that I would have thought about before. It wasn't on my typical path, but now that I'm pursuing this, I see the importance of having done that. And I kind of was semi-aware of it, but because it wasn't something that was forefront in mind, it's not something I would have considered before. And the iCore stuff, it really has been great. When I first started at BU and I had some of these initial ideas that are leading to where I am now, I thought about tech disclosure then, but the feedback I was getting at the time was that the BU support for that wasn't so great, and maybe it wasn't worth the effort given where I was. Now that I'm pursuing this, I've actually found the resources to be excellent. iCore is there. There are lots of other programs, and the resources that BU offers, they're there to help, and they want to help. They want to make it all successful. So it's really been a pretty fun process so far for me. And the last thing I've learned is that you need a champion. I'm not going to give up my day job to make this work, but Nick is going to give up his day job to take this on. This is going to become his day job. And without that, I don't think you have much hope of making a successful commercialization process. So with that, I'd like to turn it over to you, Ross.
All right, thanks a lot, Sean and Nick. I'm actually going to be joined by, I'm going to be joined by James here. So we'll do, in fact, a joint presentation. So let me just share the slides. One sec. All right, well, pleasure to meet you all. My name is Euros Kuzmanovic. I am a biomedical engineering PhD candidate and the CEO and co-founder of Biosensate. And I'm joined by my PI here.
Hello, hi, James Galligan. Nice to see you all again, and a pleasure to be here to talk about the work that we're doing with Biosensate.
Yep. And so I'd like to share, our story of going from academia to startup. And so just to introduce ourselves real quick, this is James and I doing a bit of a photo shoot together. But for myself, I really started at University of Illinois, Urbana-Champaign, where I studied biology and minored in chemistry. And so I knew I wanted to be at the forefront of research. And so I joined a chemical biology lab there. But towards the end of my undergrad, I really had this dreaded thought that my research would simply end up in a few lines in a textbook. And so I really didn't like that image going forward and thinking about a PhD. Around the same time, I joined a synthetic biology competition called iGEM, where we were forced to think about solving the world's largest problems in healthcare or agriculture or others, and to do so by engineering microbes. And so it was really in this moment that I gained a passion for doing applied research and saw that we could engineer microbes to tackle these issues. And so I pursued that further, becoming a yeast metabolic engineer. And then during a PhD at Boston University, where basically I'm engineering these novel biosensors with James to create impact. And so through this work, we were able to start this company called Biosensate. And so I'd like to introduce James. So please go ahead, James, talk.
Sure, Euros, and you can just put up the entire timeline of the thing because I can summarize sort of my background. So my background, I started out in engineering, spent a good time being an engineer, pure engineer, then took various trips through biology, ending up, for example, at the Broad Institute, finally finding my way here in this wonderful department, biomedical engineering, where the goal has been all along to bring engineering and biology together with my wonderful colleagues. And the reason why we're here on this call is about 10 years ago, as was also said by Sean previously, doing the research, making a contribution to science, having the papers, that's all fantastic. It's great, although, you know, arguing with reviewer number 3 is not always the most fun. What really became a driving force about 10 years ago was to have an impact beyond the papers. We really wanted to do something, being an engineer, building something that people will use and could be useful. That really, one day I woke up and realized I wanted to get back to that because I really, that was sort of a passion. So this idea of taking this background of both engineering, genomics, microbiology, computer science, and bringing it all together with the amazing folks in the lab and my amazing collaborators who are on this call, Mark, Doug, Kathy, Karen, and doing something that could make a real difference. That was the driving force. And the real difference, as Euros is going to describe, and I'm going to talk more about my relationship with Euros in a second, was to solve a fundamental problem at the interface of engineering and health, which is measuring our health. Euros is going to really talk a great deal about that. And I want to say what has made this journey so fantastic is both the ability to wake up every day and think that we're going to do something real, have a real impact. The collaborators that we work with across many different disciplines, making a difference really required working with people from the range of microbiology and biochemistry and chemistry through to engineering and device design. But most importantly, finding that passionate CEO in Euros. When we started this, it was really a question of who are we going to find to lead this. And that's where Euros, I think you're going to see, really has taken the horn. I've been tremendously impressed with both of his ability to know the technical work, of course, but his ability to take things and now think of it in a business sense. That's really been why the magic ingredient in terms of making this be as fun as it has. So he'll spend more time talking about that, but that's a little introduction of what's been motivating me.
Thanks a lot, James. I really appreciate those words. So I will move ahead and talk about one of my personal objectives. So Rana always asks his mentees, you know, what their personal objectives are, not really of the company or the tech itself. And for me, it's I think well captured by this image here. And so in the US, we spend disproportionate amounts on healthcare as citizens in comparison to similarly developed countries. And I'd really like to create impact in this space and change this dynamic. I think the government hasn't done enough to address this. And I'd like to do it my own way through the technology which we're trying to bring to market. This disparity primarily affects middle and low income class groups as well as minority groups about who can get access to healthcare. And so I think it's a huge problem that we need to address. What's the problem specifically? Well, infertility is a growing national and global problem in not just in the US, but around the world. And specifically in the US, though, there are almost 4 million women unable to get pregnant after 1 year of trying. And the primary reason for their infertility are really ovulatory dysfunctions, which means that they don't ovulate every month or that they ovulate irregularly. And so at Biosensate, so that's the problem. What's the need? Well, we talked to a number of customers in this space, and basically there's a group of women just like Lisa who have been trying to get pregnant for a number of months, some even for a number of years. And as they're struggling, they're getting more and more proactive about the issue, and they're using, for example, ovulation predictions test strips. But they're really frustrated by their lack of accuracy and their inability to confirm the underlying problem. And so when we think about the need, it's really for these women to assess their current fertility state. And because I mentioned ovulation is really the number one problem, the biggest cause of infertility for women, it's really to check ovulation. And fortunately for them, progesterone levels can do so quantitatively and very accurately. And so this is just basically a list of needs for a progesterone test for these women.
You're about done, Euros.
Right. Okay, sorry, Rana. Just real quick. So what's the idea? Well, we're bringing the MySense to the market, which will allow women to read their progesterone levels from blood quantitatively and in real time. And so just listing a number of value propositions here, but where are we in this timeline? I am running out of time, but I did want to show a visual representation of all the work it took last year to get us to where we are. We've raised enough money to basically execute on technical milestones. And so that's what we're doing at the moment. Just last slide, my learnings. I thought it was captured well by this comic here, where the president is looking at these plummeting sales and he's asking his VP of sales, what's going on? And the VP goes, the sales team did all they could. So I'd have to say the blame must fall on the consumer. And so, it goes to show that you can fall into this trap thinking that you created a wonderful product, but it's really the consumer's fault. The one change I have to make here is that the technical team did all they could, meaning the academic team in our case did all they could. But really what I learned is that no matter how innovative or impressive the technology really is, it doesn't matter unless you're satisfying the needs of the consumer. And the final lesson that I learned is that it's really not a sprint. It's a really long marathon, this whole growing a startup. And so it's a very long journey, which can feel lonely at times. But really, it's been a wonderful journey surrounded by a lot of positive, intellectual and encouraging people. And so very happy to be doing it. So thank you.
Thanks. Okay, Josh.
You all see my screen?
Not yet in presentation.
Yep, how's that?
There you go.
All right, hello everyone. My name is Josh Jabor. I'm working on a spin out of novel magnetic sensor technology. I am a founder of Gradient Magnetics, as well as a postdoctoral associate, where it's really, I think, the latter that comes in the primary role right now, as I just finished up my PhD. And I'll be presenting on behalf of myself and Professor Bishop, Professor David Bishop, for those of you who don't know him. So about the champions of this technology, Professor Bishop and myself, I call myself a developing scientific entrepreneur. I think it takes some of the pressure off to say developing, because we're really, Euros, myself, Nick, we're adding kind of a whole piece to our identity here. We maybe are working on being scientists, trying to become accomplished scientists, but we're developing scientific entrepreneurs. And my primary objective is to bring technology from my PhD thesis to the market. I'm remaining adaptable on that objective because there's a lot of ways that it can go. A lot of things can change based on the need. But while learning valuable transferable skills in entrepreneurship, because that helps me in my career be adaptable. Professor Bishop's objective primarily is to enable a dynamic future for students in his lab, and as well as to bring novel MEMS technologies to market, as he's done for his long distinguished career, as you can see below. So I've now had about 10 plus years experience in developing electromagnetic sensing technology, and this is on a consistent trajectory for me. And you can see that for Professor Bishop, this is also on a consistent trajectory of bringing MEMS technologies to market. So what's the need? So the need, or wait, maybe I missed, did I miss it? No, okay, there's the next slide. So the need is really kind of on the application side. We're looking for human needs. And there's kind of two main people that I look at right now for the cardiac side of our application. There's emergency room physicians. And with our technology, they could diagnose emergency room chest pain, and this will enable physicians to rule out emergencies and enable earlier and safer discharge of patients. So they right now have a complicated ability or a complicated challenge of ruling out patients who are having emergencies, right? From a cardiologist perspective, they're looking to predict coronary artery disease outcomes And we're looking to enable them to do this more accurately and to treat patients who fail stress tests and require cardiac catheterization. Our channel for this is that we're planning to either license or make and sell product to existing companies that are actually serving these customers directly. So we're kind of more of a B2B business, or selling to a business at this moment. So what's our idea? So if you look at the image on the right, it kind of describes a lot of what the problem is. The problem lies in magnetic sensing technology right now. And this is the way that people are conducting magnetic sensing of things like, the magnetic fields of the heart, or magnetic fields that can surround other organs or something like this. And what they have is they have magnetic sensors inside a large container that is liquid helium cooled. So it's at low temperatures, requires lots of sophisticated equipment, and it's all inside a magnetically shielded room with complicated post-processing and such. The shielding is required because there's electromagnetic interference from things in city infrastructure and the like. Our idea is to get rid of all of that. So we invented a magnetic sensor in our lab, and with our sensor, customers will be able to expand into portable and wearable biomagnetic imaging markets. This is going to save hospitals money, and it's going to enhance patients' quality of life. It's going to, at the end of the day, our sensor is going to be embedded in products that solve those problems that are being faced by emergency room physicians and cardiologists. And so here's a little bit about our timeline so far. And so I highlighted a few things here for those of you who are keen enough to be looking for distinguishing parts between funding, technology, and business development, because that's how we're breaking up our time at this point. So we're starting a couple quarters back here in Q2 of 2021. And so at this point, we had discovered our first sensor proof of concept. It was a eureka moment in the lab. It was an exciting moment. And after that, we talked to the tech transfer office at Boston University. And because our idea not only had significance in the scientific realm, but also it had commercial potential, we won the BU Ignition Award. And that allowed me to fund my postdoc here and to continue developing the technology toward commercialization. Then at the end of 2021, we developed our next generation sensor, which enhanced its commercial capabilities as well as its resolution. And with the help of Rana and other business mentors inside of BU and outside of BU, we've identified our need, our target market. We're staying flexible on it and adaptable to make sure that we're serving a true need. We've done some entrepreneurial education, and then we won our first academic pitch competition, which perhaps validated our ability to at least do some business planning as scientists and as scientific entrepreneurs. Then in the last quarter of 2021, we actually obtained our first detection of cardiac magnetic fields, which is no small challenge with a novel magnetic sensor or any magnetic sensor. And then with the help of BU, we've been building our IP barrier around our core technology. And then I also participated, like Nick, in the I-Core Spark program to enhance my understanding of a product market fit and to fit the product I'm trying to develop to a real need in the market. And so here's where we are now in Q1 of 2022. We're working on now optimizing our sensor for these cardiac applications now that we're on the map in terms of detecting those fields. We won a research grant to hire a PhD student and get some more help in moving this faster. And then we're preparing some of our early studies here for publication in a scientific journal to build credibility for the idea and the project moving into a business. In Q2 of 2022, we plan to build our first prototype for the goal of de-risking our clinical proof of concept. And then we plan to build IP barriers around the application. And then finally, in Q3 of 2022, we're planning to achieve some follow-on funding from either SBIRs, STTRs, or private fellowships. And then that we're looking to help us begin our clinical measurements and actually look for some of those key needs that we've identified. And at that point, we're going to need to be flexible as well in terms of what we find. So what's my primary learning? And this is, of course, as Rana said, as an individual in this process. Well, a lot of us are coming from a scientific perspective. Probably, I'd estimate something like 70 to 90% of the people on this call. And what I've kind of found through my mentorship here is that approaching the business with a scientific method can work for technology spinouts. And this is a lot of the preaching of the NSF I-Core team as well. You don't have to transition and become a businessman right away or a businesswoman right away. And so, this is some of my probably preconceived notions about business that are wrong that I'm going to correct for you in a second. But there's some hand-wavy parts of business planning that can feel like they lack credibility, especially when there is always a right and wrong answer in science, right? There's a ground truth. It's either there or it's not. However, your training as a PhD scientist can actually make you a very effective scientific entrepreneur. For example, whether your business viability would rely on 100 technical customers buying an expensive scientific tool, or 1 million general users integrating a part of your technology into their daily life, you can do research on those items, right? And you can prepare arguments to defend those estimates. So at the end of the day, nothing should be hand wavy, even though there may be a higher degree of uncertainty in those estimations. So to be successful here, we can apply the scientific method. You can develop hypotheses about your markets, about your needs. You can develop methods for gathering information, and then you can organize those results and prove it to yourself that there's a need that exists that you're chasing. And in the future, then you're going to need to prove that to your investors. So thank you for your time, Rana. And now George, I think.
There you go.
All right, can you guys hear me? Okay, so who are we? So I'm George Tabul. Did my PhD, I think, over a decade ago now. So in biomedical engineering at BU, where I did my senior project in Selim's lab, and that kind of launched me into our PhD program that kind of culminated in the technology that we started the NanoView Biosciences on. And during my time at Selim's lab, I met David Friedman, who was a postdoc at the time, who actually was the first one to launch the company off of the ground. And then I joined him later after my postdoc. So why did we, what was our objective, or my personal objective? So I knew academia was not for me. I saw the arduous path of becoming a PI. So, but I wanted the independence to pursue my own ideas. And so startup was really attractive to me. And it was the right time to take the risk. I think someone else said this is, as a grad student and postdoc, you don't really make much money. I don't know how much they're paid now, but back then it was very little. So if you want to pursue science and not make a lot of money, I think startup was the next best thing other than pursuing a PI. So that was my objective. And one thing I want to give a caution on independence is once you raise VC money, some of that independence is gone. So enjoy the SBIR and non-dilutive funding time to really hone in your idea and understand where you're going with your company before you raise VC money. Because once you raise VC money, it's really adding fuel to the fire, and you really want to make sure you know where you're going. So what's the need? So our customer is either a researcher, either in the corporate or academic setting, and like to characterize the composition of biological nanoparticles. And those biological nanoparticles can be applied in different areas. So if you're developing drugs or therapeutics, as you guys know, the COVID vaccine is based on a lipid nanoparticle delivery. You need to be able to characterize those biological particles. And also, if you're developing a diagnostics, you can look at biological nanoparticles as biomarkers. So our XView platform provides multiple data points like size, protein composition of these biological nanoparticles in a single, easy-to-use assay or measurement. And this was based on the SBIRS technology that was developed in Selim's lab. So what we did is leverage interferometry, which Selim is a pioneer in, to enhance signal coming from biological nanoparticles that we capture on the surface. So we're able to leverage interferometry to measure the size of these nanoparticles that is typically you can't really do with conventional bright field microscopy. And also at the same time, it enhances fluorescence microscopy, which is usually used to tag different proteins or biomarkers on these nanoparticles. So at NanoView, we took this technology and then we made a product out of it. So here, this is our reader, and we sell with that reader different assays and kits to address different market needs. So we took what is currently a really arduous workflow and compress it in a much simpler workflow where people can get data much faster and be able to advance their programs, either research or drug or biomarker programs much faster and more cost efficient. So where are we now at NanoView? So I'll go through our timeline. So like everyone else here, we started with iCore while I was a postdoc at Selim's lab, and David was launched a company, and there where we kind of honed where we're going to apply our technology, what was the great market fit there. We applied for a bunch of grants to kind of hone the ideas and kind of do development. That was 2015. Then once we've defined the application area we were interested in, we filed more patents and raised our venture money to productize and launch the product. Basically, we'll launch our product in early 2019, which is the reader I showed you guys with different kits for different application spaces. And then in 2020, basically we had about 30 employees and we raised our second round of funding. And right now in 2021, we closed with around 100 plus customers that are using our platform for different application spaces. And right now we're really positioned to more in the growth phase. We've understood really our market product fit. We have great amount of customers using our product. And now we're looking to accelerate that growth and applying our technology in more applied fields like liquid biopsy for cancer diagnostics or and right now we're raising more capital to kind of fuel our growth, basically. So learnings from starting NanoView, I think having a co-founder is key. Like I think Euros mentioned that it's not a sprint, it's a marathon, and it's a roller coaster of a marathon. There's good times and tough times, so it's always good to have someone to lean on. And it's always good to find a co-founder that has complementary skills. So that helps you kind of move things forward faster. But also what I mean by complementary skills is also someone that will do things that you don't want to do either. So basically everyone gets to do what they like and contribute effectively to the growth of the company. And with that, I think that's my last slide and turn it back to Ron.
All right. Fantastic. Really fantastic. Thank you all. And it was a lot of work putting those together. Actually, I appreciate you guys entertaining my questions.